for waiting. Thank you for waiting, everyone. Welcome to IMBF seminars number 7. So I will make a notes for now, like the YouTube is not working. So we'll just upload the recording to the YouTube. So I will just encourage everyone to use the Zoom for these webinars, and we'll upload once it's finished. So, good morning, everyone. Welcome. I am Ita Wulandari, and I will be moderating for today's webinars. And it is number 7 webinar series with our speaker, Dr. Suliskania Nurfitri and Miss Kanita Utaipan. And we'll talk about the ocean deoxygenation and upwelling in the marine biogeochemistry. And we'll divide this session into 4 sessions. First, we have opening remarks from Professor Aan. And next, we are doing the first speaker, Dr. Sulis. And then the third one, we'll have Dr. Kanita Utaipan will talk related to the hypoxia. And then the last one will be Q&A session. And then without further ado, we'll start the opening remarks by Professor Aan. And everything's yours.
Thank you, Ita. Good morning, everyone. And first for all, I would like to thanks for the coming in this webinar, and especially for the speaker today, Dr. Suliskania and then Ms. Kiki, that able to join us and will share their research about upwelling role in biogeochemistry and also for the deoxygenation or hypoxia process in the coastal area. And also thank you for attending this webinar for all of the audience, and we hope that this webinar can give us insight for the marine biogeochemistry research. And without further ado, then we can start with the this webinar, and I would like to thanks again for everyone. Thank you very much.
Okay. We'll continue with our first speaker. Dr. Suliskania Nurfitri is a lecturer in the Faculty of Earth Science and Technology at Institut Teknologi Bandung. Her expertise lies in oceanography and marine ecosystem modeling. She holds Bachelor of Science in Oceanography and a Master of Science in Earth Science from Institut Teknologi Bandung and a PhD in Oceanography from University of Hamburg. Today, Dr. Nurfitri will discuss the role of upwelling in marine biogeochemistry. Okay, the times and the screen is yours.
Okay, thank you, Miss Ita. Can everybody hear me? Okay. Good morning, everyone. And first of all, I would like to thank Professor Aan for giving me the opportunity to share what I'm doing in my research. Okay, maybe I will start share my screen. In a second. Can everyone see my screen?
Yes.
Okay, thank you. So I think I have 20 minutes.
Yeah, you have 20 minutes, but please make it full screen because in the moment, great.
Okay. Yeah, so maybe, okay, thank you, everyone. So today I would like to talk, maybe it's a little bit different with the title in the poster. Today we'll talk about the understanding the nutrient distribution in the upwelling system using numerical models. So I will start with the outline for today's talk. I will start with the introduction, maybe talk about how the models work, and The second part is the ecosystem model approach. I will show some of the approach, especially for the ecosystem models. And the third part, I will show one of the models that I have used before, the EcoHam or Ecosystem Model Hamburg. And in the last section, I will show also the study case in Arafurasi upwelling system. Okay, I will start with the introduction. Okay, so maybe some of you know how to simulate the hydrodynamic in the oceans. I will start with the physical models. Here in the physical models, we have the established equations such as a hydrodynamic equation, which explain the conservation of mass, momentum, and energy. So this equation is quite established, so which means that we use, so everyone who's doing hydrodynamics modeling will use the same equation. This is the well-known and accepted laws, and in this way, from the equation, we develop the numerical models. So as for the biology, it is a bit different or more complex. Here in biology, there's no generally accepted laws and also large complexity, and there's no established equation. So in this way, the first step, we must develop the conceptual model first, and then we develop the numerical models. So the step is a bit complex rather than the physical part. So marine ecosystem models and biogeochemical models share a similar structure. So sometimes we call it as a marine ecosystem or biogeochemical. So these two models are similar but differ in their scientific question. So it depends on how or which aspect that we want to focus on. And these models account for living organism and non-living organic, and also nutrients and other components. Yes, such as phytoplankton or zooplankton. I will show you the example after. And as for the physical ocean model, here hydrodynamics model describe ocean physics, such as temperature, salinity, and also the external conditions such as which affecting the ocean, like wind, solar radiation, and etc. So to simulate the ecosystem model, we need the hydrodynamics model. So because we need the forcing such as, for example, temperature condition and also ocean current. Okay. So for conceptual models of the marine ecosystem, there are two types. So first, based on trophic levels. Here, the example of the trophic level conceptual models. In this way, we focus only on the food web. So there's a clear distinction between trophic levels from the lower trophic level up until the highest trophic level. This is the trophic level conceptual model. And the second type is the elemental fluxes. So the figure on the right shows the scheme. This is the example of the elemental fluxes conceptual model. In this way, we focus only on the elemental pathways with the simplified food web. So in this type of models, we only focus on the flux of elements such as nutrient or carbon. So in this way, we must simplify the key species. Here, for example, a phytoplankton, we see phytoplankton as the bulk phytoplankton in this type of models. So we don't differ this into different species. Yeah. So I will talk detail about this later on in the EcoHam section. Okay, and for the approach, so in general, there are 5. I only show the most general ecosystem model approach. So first one is prognostic versus diagnostic. And the second one is individual versus compartment, Eulerian versus Lagrangian, empirical versus mechanistic. And the last one is steady state versus time dependent. So I will talk in more detail later on. And in ecosystem models, we describe state variables, which the variables that evolved with time. and we describe the process that link these state variables. Okay. So first approach is prognostic models. So in this approach, we include time dimension. So in this way, we can predict the future of a certain component of a system. At least one of the state variables is prognosis variables. Here, for example, like the time rate of changed biomass, growth rate times actual biomass. Yeah. So this way that so the time rate, because there is time rate, which means that this variable is changed over time. So there are three types of models in prognostic. We can do a forecast, like a prediction. We can do also projection. This is similar like forecast, but based on the present data. And the third one is hintcast. So this is like we simulate in the past. So it's like the representation of the past state. As for diagnostic, so this is there's no time evolution. Here, for example, like production equal to catch plus predation plus net migration plus biomass accumulation and other mortality. This, for example, input variables like biomass, production ratio, consumption, or biomass ratio. So this is the key points of prognostic and diagnostic, and it is because of time dimension. Yeah, there's in prognosis there's time dimension, as in diagnostic models there's no time evolution. Okay, the second approach is individual-based and compartment. In individual-based model, or we call it IBM, this is focused on the individuals, or often grouped into one entity. We call it as agent. Yeah, so the concentration of organisms are the result of the distribution, number, and size of individuals. And for compartment, we focus on ecosystem compartment. Yeah, this is and we compute changes in the concentration of the compartment in time and space. Yeah. So what I did previously is compartment model. This is also included in EcoHam. I will show you later on. And the third type is Eulerian and Lagrangian. So both Eulerian and Lagrangian approaches can be used for observations or models. As from Eulerian is focused on continuous fields, where Lagrangian approach examines individual particles. So in Eulerian, we focus on distribution of organism as the continuous field, but in Lagrangian, we focus on the individual. So organism treated as discrete individual with certain properties. And these properties or individual transport with flow feet through time and space. So change in properties like behavior, life cycle of individual can be directly considered. This is for Lagrangian approach. And empirical versus mechanistic. So for us, for empirical, this is the approach which arises from experience and discover experiments. We can also use statistical exploration of correlation with environmental variables, and of course, many environmental variables are very difficult to resolve cause from effect. As for mechanistic, this is explained by laws of nature, include aspect of organism function and behavior. And the last type is steady state and time dependent. And steady state related with time, there is no change over time. So which means that the system reach the equilibrium state. As from time dependent, there's change over time, change of the system over time. And this is also more realistic because everything flows. Okay. Now I'll move to the EcoHam. This is the example of the ecosystem models. So according to the previous type of models, EcoHam is categorized as the compartment models, and we focus only on the elemental cycles. So this is so the figure on the screen shows the EcoHam schemes. This is show how the model component interact with each other. So in general, there are two types of phytoplankton considered in EcoHam, diatoms and non-diatoms. And also there are two types of dissolved organic material, labile and semi-labile, two types of zooplankton, micro and meso, different from the size, only from the size. Also two types of detritus, small and large detritus. So there are a lot of arrows which show the flows from one component to another. Here, for example, black line show the predator-prey interaction. So which means if the arrow moves from phytoplankton to zooplankton, it means that zooplankton eat phytoplankton. And also this one, bacteria to zooplankton. And the green line shows the fluxes of non-living organics, like this one, the green line. And the red line shows the nutrient fluxes. This is the nutrient considered in aquam. There are 4, silicate, phosphate, nitrate, and ammonium. The blue line shows the flows of oxygens. And as for the dash line, blue dash line shows the carbonate system. And there's also the sediment system in the EcoHam also. So this is the simplified scheme of the EcoHam. So this depends on user which aspect that we want to focus on. We could if we want to focus for example on phytoplankton dynamics here, for example, we can simplify the carbonate system. So it is depend on which research question that we want to answer. Okay. So before we doing any modeling, we start with the partial differential equation, which describe how the every state variable evolve over time. This is the example of one state variable, P, or phytoplankton. So P is the function of X, Y, Z, and T. X, Y, Z indicate the space, 3-dimensional space, and T indicate time evolution. So if we derive P with respect to T, we have these terms, and with chain rules we have the complete terms. So the first term show the derivative to the X direction. Yeah, the X term means indicate U, or U we call it as the current component in X direction, or in the west or east direction, V in the south to north direction, and W shows the vertical component. Yeah. So this is the simplified term. We call it as advection of P, or advection of phytoplankton. This is the terms. So in the complete terms, in the physical part, we have the advection term and the diffusion terms. Yeah, so there are two processes in the physical part, which is advection and diffusion. So this is the complete term of the physical part for the change of phytoplankton. And next, we add it with the biological part, this one. So in ecosystem model, we have two parts of change. So the first is change with the physical process, and the second one, the change because of the biological process. So the physical parts, as I mentioned earlier, relate with advection and diffusion. And as for the biological part, this is related with the scheme that I showed before. This is related with the source and sinks. So everything with the biological process, with chemistry process, we included in the biological part, like this one. So source and sinks, this is the example of the source and sink for phytoplankton. Of course, we have a more complex equation in the EcoHam, but this is the most simple term for phytoplankton, like we have source and sinks. Source coming from the growth rate, like mu, and sinks from the mortality rate. Of course, we can add more other terms like grazing, because grazing by the zooplankton, or maybe other part. But this is the simple equation for the source and sink of phytoplankton. Okay. I still have time, right? 10 minutes, okay. So next I will move to the study case. This is the study case in Arafurasi. I'm sure that most of you know where Arafurasi is. So Arafurasi, located in the eastern part of Indonesia. This is the Banda Sea, and this Arafurasi, this is the New Guinea or Papua. So the figure on the left shows the satellite-derived chlorophyll A concentration, which show that there's elevated of chlorophyll A concentration during the southeast monsoon, which occur in around June, July to August. So during this period, there's the phytoplankton blooms, as I mentioned earlier, related with the upwelling, which observed in this period. This is the model findings by Kempf in 2015. So the research by Kempf shows that there's the undercurrent-driven circulation in this area, which bring nutrient from the Banda Sea to Arafurasi. That which bring a lot of nutrient and cause so that's why the chlorophyll A is high during this season. Okay. So to simulate the condition in Arafurasi, there are two models that I use. First is HAMSOM, or Hamburg Shelf Ocean models. This is the hydrodynamic part. So HAMSOM was run first, and from HAMSOM we get a lot of variables or input, and sometimes we also call it as a forcing that used for the EcoHam. So these two models were run separately. So HAMSOM was running first, and then I use the output from the HAMSOM, such as temperature, also salinity, and I think the most important would be the ocean current to be the input for the EcoHam, the ecosystem model Hamburg. So of course, in another model, we can also run hydrodynamics and also the EcoHam simultaneously. So in this way, we call it as a couple model. But in other method, we could also run the hydrodynamics first and then the ecosystem model after. This is the model design for the hydrodynamics, which also the same for the ecosystem. This is the domain for the models, which cover also the Banda Sea, because we want to see how the nutrient coming from the Banda Sea to Arafurasi. So this is the domain for the models. And the contour here shows the depth or bathymetry of the area. Okay. So the horizontal resolution for the model is around 7.4 kilometers, and we have also 45 vertical layers, and the thickness varies from 5 to 500 meters in the deep layer. As for the forcing for the hydrodynamics model, there are 4 forcing, and at the surface we have meteorological forcing for UNCEP, like wind, also solar radiation and temperature. And also we have temperature, salinity, and sea surface height along the lateral open boundary. So this is what we get from MPOM. This is the Max Planck Institute Ocean Model. So they have the global model, and we take the data at the open boundary. And also at the open boundary, we prescribe tides. This is, we get it from the TPXO global tide solution. And the fourth forcing, we also include river. So the river we get from water gap global hydrology models. This is for the hydrodynamics part. Yeah. And for the ecosystem, there are some input. So first, as I mentioned earlier, I need the input from HAMSOM. This is the 24 hours averages of hydrodynamics fields. This includes the temperature, salinity, and also the ocean current, the ocean, and also the height of the sea level. And the second one, we also use shortwave radiation from IMBF for the forcing at the surface. Atmospheric nitrogen deposition. This is for the input in the nutrient cycles, and also river. And for the ecosystem model, we need two types of river input. First one is fresh water. This is we also get it from the water gap, the same as HAMSOM, and also nutrient loads. So there are two types of river input: fresh water and how much nutrient that brings from the river. And for the lateral open boundary, we also we also prescribe all the state variables, and because it's a bit difficult to get the observation data, so that's why we use the World Ocean Atlas and combined with GCOMS. This is the Global Coastal Ocean Modeling System. So in EcoHAM, in every time step, we must prescribe all the state variable along the open boundary. So maybe I will return a little bit. So along this open boundary, we must prescribe all the state variables. So in this way, so it's a bit difficult to find the observation. That's why we usually use the World Ocean Atlas. So the value itself is not changed over time. Sometimes we could also prescribe it as a seasonally change, but to simplify it, we could also use the constant value over time along the open boundary. Okay. Okay, and for the phytoplankton growth, maybe I will show some of the limitation factors. So in general, there are three factors that limiting the phytoplankton growth. The first one is temperature, the second one is nutrient, and the third one is light. So to describe the limitation of temperature, we use this formulation, FT, which means the limitation function with Q10. So Q10 indicate how much the growth rate of phytoplankton change over 10 degrees increase. Yeah. And the value itself ranges from 1 to 2. In EcoHAM, we use 1.5. So it depends on which area that we're simulating, but the standard value is usually we use 1.5. As for nutrients, this is the general equation, nutrient limitation. We have nutrient concentration N, and Kn is the half saturation constant. So the limitation nutrient value range from 0 to 1. 0 means very limiting, and 1 means that the nutrient is abundant. As for light, this is the light dependent growth rate, or we call it as F bar. This is the formulation. There are a lot of formulation, but this is what I use for the EcoHAM. Yeah. Okay. Okay, 3 minutes left. Okay. So I will show some of the results. This is the result from the HAMSOM model. So we have the surface current in January and in July. So in general, yes, this one. So in general, in January, there are current that flows to the east, like this one that I show in the arrows. And in July, the currents is move from the east to the west and is much stronger. So this is the surface current that which triggers the undercurrent circulation. I will show you the vertical shows after, like this one. So in July, which is during the southeast monsoon and during the upwelling period, so if we take this one, the cross section, like this one. So this is the plot of zonal velocity of the component of west and east direction of a current. So blue indicate that the current moves to the west, like this one, and red indicate that the current moves to the east. So we can see that in the surface up until like 100 meters, the current moves to the west, and the undercurrent, like this one, moves to the east. And this undercurrent brings nitrate on the, or a nutrient. Here, for example, I show you the vertical distribution of nitrate in the same cross section in July. So if I take this one in 135 longitude and plot it over time, this is the Hofmuller diagram. We can see that in June, July, and August, there are the uplift of the nutrient. Here, for example, nitrate during this period. So in this period, that the high nitrate concentration moves or maybe brings to the upper layer. And in the same location, this is the vertical profile of stimulated nitrate in January and in July. We can see that there are strong stratification in July, like this one in the red lines. And this is the plot of two physical process. The first one is vertical advection flux, which means that the red one shows that the flux is upward. So it's bringing that nutrients from the lower level to the upper level, as the blue indicates that the nutrients is downward, so the negative flux. As for mixing, this is related with the diffusion terms. And compare with these two processes, the advection is relatively more dominant, than the mixing flux. So if you see the magnitude itself is a bit different. Like a mixing is much more small in magnitude. Okay, that's all. Thank you very much, everyone. I will look for the further discussion.
Thank you, Dr. Suli. Such a great presentation, and I enjoy your presentation. And then, if there is any other question, please put in the chat box for now, or later we have a discussion session after Kiki. Okay, for now, we'll continue on the next speaker, Kiki. All right. Kanita Utaipan is a PhD candidate in marine chemistry from Ocean Carbon Group in State K Laboratory of Marine Environmental Science, Department of Oceanography, College of Ocean and Earth Science, Xiamen University in China. Her expertise lies in linking biogeochemical process to environmental issues like eutrophication, hypoxia, and ocean acidification. She holds Bachelor of Science in Teknologi in Marine and Coastal Resources Management from Upwelling University in Thailand, and MSc in Chemical Oceanography from Chulalongkorn University. Today, Ms. Kanita will discuss the coastal hypoxia causes, effects, and solutions. Okay, for now, the time and screen is yours.
Oh, apologize. Do you able to hear me?
Yes, we can hear you.
Oh, it seemed to be I cannot.
Say it again. Oh, it's freeze. Okay, for now we'll wait until Kiki will going back. Um, okay, for now we need to wait for a while until Kiki is going back. So if there is any question for Dr. Sulis, feel free to put it in a chat box or we can start until waiting for Kiki. Any questions from audience? So I have a question actually. So since the models developed from the Hamburg University, if I'm not mistaken, is it?
Yes, from Hamburg University.
Yeah. Is there any similar models from any other location that also works related to the environmental modelings on the coastal systems that you ever been working with?
Okay. So actually there are a lot of ecosystem models like maybe have you ever heard like ROMS? ROMS also has these models for ecosystem, but for me I like for my dissertation I ever only use EcoHam. But there are a lot of models that ecosystem models that can be used. But I think most of them is developed not in Indonesia. So I don't think I haven't found any models that been developed in Indonesia. Maybe in the next few years, I hope so. but no, I think so there are a lot of ecosystem model, but for me, I only use EcoHam.
Mm, that's interesting. So with the models like using the Hamburg one, what is the tricky part of using the models? Like you are comparing with the other one or
Mm. I think the most difficult part would be preparing the input data. Because I mentioned earlier that in ecosystem models, we need much more data than hydrodynamics models. For example, like we need nutrient value, we need phytoplankton abundance, or maybe if we want to talk about the carbonate system, we need maybe the DIC concentration here, for example. So this is the very, very tricky part, I think. So that's why sometimes we use the constant value from the global data, for example, like for global data set. So in this way, we can we can do the simulation. Otherwise, yeah, we use how do I say it? Like the artificial data set. But it's a bit difficult also. But I think the most probably the most possible one would be take the data from the global data set. I think that the most tricky part is preparing input, preparing data set. I think that's the most difficult. And also, I think the computational time also, it takes very long, especially if you have very large domain. And so I run it in the supercomputer back in Hamburg. So it takes maybe 3 days or so. But if we don't use supercomputer, it will take longer. So I think that's one of the challenge if we want to use modeling is of course we need a computer, we need the resources to run the models.
Yeah, that's very tricky that we need the facilities. It's quite important for running the models.
Yes, exactly.
Yeah. Yeah, until now we
She's not
cannot hear anything.
joining.
So, yeah,
Okay.
we do not hear anything. So, anyone that have a question for Dr. Sulis, please feel free because it is like a discussion. And yeah. Oh, that's great. We have one participant want to ask. Um, yeah. I think until Kiki, hello.
Hello.
Everything's good. So for now, we have a Q&A until you're going back.
Okay, you can done first, and after that I will follow. Now I think I'm ready.
Okay. So we save for the question for later. If I'm not mistaken, Mareta. Yeah. You'll be the one the first after the for the Q&A session. But for now, we'll continue our session with Kiki. Kiki, are you ready?
Yes. Do you see my screen?
Not yet. It's still blank.
Oh, still
Oh,
blank.
yeah. Oh, yeah. It works now.
Okay. First of all, I would like to say good morning, everyone. And actually, I'm glad to be here with a good environmental and atmosphere with a friend from the Southeast Asian country. And my name is Kanitha Uthaipan. Today, I'm glad to bring you to my journey about the eutrophication and hypoxia in the coastal zone. And in here, I will generally specific to the cause, effect, and solution. And again, my name is Kanitha Uthaipan, a PhD candidate from Xiamen University, and also at a youth committee of the Coastal SOS program, endorsed UN Decade program of the Ocean Side for Sustainable Development, belong to the Xiamen University. And thematic outline today, I would like to talk for a topic eutrophication and hypoxia in the coastal zone, cause and effect, solution, and also about what is the coastal SOS program. When we talk about the eutrophication and hypoxia, everyone can imagine the picture because it occurs worldwide, that we can see the green color in the ocean coastal, or maybe sometime in the red color, and maybe another type of the color specific depend on the phytoplankton species. But this figure, it come from the upper Gulf of Thailand. Everywhere in the upper Gulf, it covered by the green noctiluca. And if you see in the figure, this is the fish. This is the sea mussel cat along the coastal zone. Contrast to the eutrophication, look green. Some people may say that it's beauty, And some people still use the coastal zone even they have the blooming. But after a while of the bloom, consequences result clearly shown that the dead zone appear mass mortality of fish and many animals along the coastal zone cover many kilometers in the upper Gulf of Thailand. And this, what is it of the coastal eutrophication and hypoxia that we can observe from eye? But if we're talking about hypoxia truly for biogeochemical, that what we're talking about? We're talking about the DO concentration dissolve oxygen deplete in the water below to the 2 milligram per liter. Some people they use the 62.5 micromole per liter. Or if you are marine chemistry, you will use the 61 micromole per kilogram. And also sometimes some people said that about 30% of oxygen dot of the unit, it belong and functioned of the 25 degrees Celsius and salinity at the 35. And this value come from the scientific committee working on the oxygen. We call the score working group number 128 that had been referenced in many papers. And this figure can show that clearly. All of the units can vary, but typically in the coastal zone, we will counting at the below 2 milligram per liter. That is the animal cannot survival or it have important impact on the physical process in the animal as well. And in here, when we talk about the coastal eutrophication and hypoxia, where we found eutrophication, mostly we will find the hypoxia as well. If you see the red dot it cover whole of the coastal zone in the worldwide. And in the contrast, in the open ocean, also found a low oxygen concentration. If we're talking both area, coastal zone, and also in the open ocean, decline in oxygen we will call deoxygenation. That it come from D, that it a down, decline, oxygenation, that it mean oxygen, bind together, that it mean decreasing or decline in oxygen in worldwide. And in here recently, paper, they said that the water quality in terms of oxygen near to you, that it means oxygen decline not only in the coastal zone, but also in the semi-enclosed Great Lake, upwelling system, open ocean, even though in the river as well. And primarily with the activator for the ocean oxygenation, we have to divide it. Firstly, we have to divide into area. One in the estuarine and coastal zone, and secondly that in open ocean. These two areas, when the oxygen decline, they have the different process. And in here today, we will focus in the coastal zone where it connecting with the river and estuarine, where the intensity of the nutrient come from the river. And when nutrient go to the coastal zone, stimulate the eutrophication in the surface layer and occurring hypoxia below the stratification. In contrast to the open ocean, in the open ocean, in the different processing, but it's same situation that it induced by the ocean warming and human activity as well. And in here, what is the interaction of the eutrophication of the hypoxia and eutrophication? When we studying the coastal eutrophication, that we have to look back to the watershed, we cannot only concentrate in the coastal area where the eutrophication and hypoxia occurring, because in this area that is the final result we saw but the original of the source and a problem occurring in the watershed. So in this study, we have to emphasize from the watershed, go to the estuary, and also go to the coastal zone. And in the sum of the connecting area, we connect to the upwelling system, go to the open ocean. As I mentioned before, the watershed area is important in terms of the source of the nutrient, nitrogen and phosphorus, and sometimes it stimulated and intensified by organic matter. And in here, when we go to the in the estuary zone, in this area combined by many and intensive of the coastal activity, because over 10% of the worldwide coastal area is facing by the coastal eutrophication and hypoxia due to the nutrient input, not only from the coastal zone, but it from the upper shed and also from the groundwater discharge as well. And some of the system low concentration of oxygen supported from the upwelling system. And so the coastal zone where we concentrated at the point that we saw the problem, that it because of the eutrophication stimulated from assimilate the nutrient from the river, and also have the some from the organic matter from the organic matter from the estuary, and where we call organic matter from the terrestrial. But if it bloomed by the phytoplankton, we call it organic matter in the marine system. And this is occurring in the surface layer where the plume spreading to and bring the nutrient to. Below the plume, or we call the stratification, when the phytoplankton die, they will go and sink down, and this is consequences result for the oxygen consuming by the microbe, and oxygen will be depleted. It contrast to the upper layer that where oxygen saturated. And in here, one of the global model when they're studying the coastal eutrophication and hypoxia, people will think to the Mississippi River and also Gulf of the Mexico. This is the whole area, the Gulf of Mississippi River, and whole of the area produce nutrient and cause of the eutrophication and hypoxia in the coastal zone. It lack one of the larger dead zone in the worldwide as well. And why it's important? Why eutrophication is important? Because it's quite common. Everyone can heard, if you are marine scientist, everyone heard about this situation. Everyone know about eutrophication and hypoxia, but actually the eutrophication and hypoxia, one of the problem that it's difficult to tackle and solve this problem, that it's not easy, even though in some country they are developed country, and this is also challenging for the developing country, such as in the East Asian and particularly in the Southeast Asia. If we see development in the 1969, some of the field research observed in the developed country and no science study in Southeast Asian country. But after that, in 1990, around some of the Southeast Asian country appearing, and more frequency, and currently more than 600 hypoxia related to anthropogenic cover the coastal worldwide, and also covering more than 245,000 kilometers. That is large if compared to the coastal zone. There are some of the point concerning that is the developed country appearing before developing country, and now this is the situation is ongoing in our region. That is why we have to pay emphasis on the eutrophication and hypoxia before they go to the tipping point that we can not recover our ecosystem. And in here, why in each Asian country it's important? Because in this area, most of the human footprint and also nitrogen load is concentrated in this area, and also high concentration of the chlorophyll concentration along the coastal zone, and also the found more and more hypoxic site. And in here, when we talking, as I mentioned before, why there are different development between the developing country and developed country, but they are in the same story and in the same trend. Firstly, they stimulated from the agriculture to the industrial revolution, totally that is a civilization of the socio-economic force, because the agriculture support to the population growth and need fertilizer consumption, and support agriculture system support to the urbanization and also industrial. So where we are going now in the Southeast Asian, we are going in this trend, and we hope in the future we're not going to the uncontrolled system, but hopefully before we go to that tipping point, we can control some of the nutrient reduction for the eutrophication, harmful algae, and also hypoxia in a country, in Asian country. And in here, we clearly see that when the eutrophication and hypoxia occur, biodiversity is lost. And if we are marine scientists, particularly in marine geologists or marine chemistry, we're concerning about the cycling of the carbon, nitrogen, and phosphorus, because that is the kind of the feedback to the global warming, and carbon release to the carbon release to the atmosphere again. So if these two simple cases occur together, that it means we lost the security of the food consumption and also impact to the economic loss. And finally, we will lose the capacity to management and also mitigation if we can not control, and if we let our ecosystem to the uncontrolled conditions, so ecosystem will be disrupted in the future. And in here, this is a simple diagram show about the triple threat between the eutrophication, hypoxia, and ocean acidification. Firstly, I would like to show you this diagram. This side, it comes from the river. This side, it is ocean. And when the hypoxia and eutrophication is occurring, that it then need the stratification. Stratification, it comes from the difference of the salinity and also temperature, that it cause of the boundary. The surface cannot mixing to the bottom. And in the upper layer, when the nutrient come to the coastal line, the carbon stimulate together with the carbon dioxide consumed from the atmosphere, change to the organic matter form, and then plus oxygen concentration. If organic matter sink down, that it means phytoplankton die, they go down, that it means when they go down, this organic matter will be consumed by the microbe and need oxygen. That is why in the bottom layer, oxygen deplete, because when they want to degrade the organic matter, they need oxygen. During the same process, when the oxygen decline, carbon dioxide also going back to the water again and go up to the water and go to the atmosphere. That is why the loop of the eutrophication, hypoxia, ocean acidification, and goes to the global warming again. This is quite complex system, but primarily that is a surface eutrophication and bottom hypoxia and ocean acidification. And in here, one of the case study in my country, that is Thailand, we can see whole of my country, in the Gulf of Thailand, many eutrophication occurring, particularly in the upper Gulf of Thailand. In the same area, we found the hypoxia, and in the same area, we found ocean acidification as well. And this is the trend of the eutrophication. We found the red tide increasing at least one time a year. And in the same time, in the upper Gulf of Thailand, the trend of the eutrophication, we found that. And in the same trend, we found decline in the oxygen concentration and found more the dead zone or hypoxia zone in the upper Gulf of Thailand. So when we want to study eutrophication and hypoxia, as I mentioned that if we look at only the sea, we cannot tackle and we cannot reduce the nutrient or activity from the land. So when we frame the study, we have to emphasize both of the land and to the sea. We call the continuous system, fresh water to the ocean. And in here, this is the side flag of the upper Gulf of Thailand. They are consisting by 5 rivers, and also we have investigated in the upper Gulf of Thailand. And how we investigate, as you see, the eutrophication and hypoxia, it looks really simple for normal people, but we are scientists. We have to look in the holistic view, and we have to observe from the land to the atmosphere, biodiversphere, in terms of biogeochemical cycle, and also ocean cycle, that it meets circulation, it need water current, it need atmospheric, it need, and absolutely in the land, socio-economic also need. And in here, from my study, along the development of the country, we can see clearly different color and different A, B, C, different year in terms of the land use. And we can see clearly in 2000, whole of the area mostly consisting of by agriculture, only few concentrated of the urban, and more developed in the recent year. And what it means, deforestation change to agriculture zone, from the agriculture zone change to the urban land, and from the urban land change to the multiple land use type, but primarily that the deforestation change to the agriculture and change to the urbanization. And after change to the agriculture, that it means more input of the nutrient concentration, both of the nitrogen and phosphorus. And in here we can see clearly, after we see the nutrient increase, that it means water quality also degrading. And many rivers decline in terms of the water quality. And clearly we can see the low concentration of oxygen as well, not yet only in the river, in the estuary. As I mentioned before, we have 5 estuary, also see the same trend, and also found the hypoxia in the upper Gulf area as well every single year. And in here, why it occur only in the lower reach of river? If we translate whole the picture to the simple diagram, from the 5 river, we can see different pattern of the development. In the upper, the high biodiversity of the cell purification, and so high velocity, that it means high stream flow. So oxygen can exchange between water and air, but it come to the lower rate, it very intensive by the human activity, that it why cell purification it quite low. And furthermore, that it is nutrient accumulation from the upper to the lower, it increase and increase. Combined with the physical processes, it the cause of the oxygen deplete in this area. And as I told you before, that the eutrophication cause of the oxygen decrease, but in the same time, along to the coast, along to the oxygen decline, also found the ocean acidification. If we look back to the pre-industrial, the oxygen concentration and pH concentration should be decreased in the range of the normal range at the egg, pH at the egg. But now, currently, we found that during the eutrophication and hypoxia, the pH concentration going down and going down. And how about when we predict to the future? That is the trend line of the future. That is what it means in the future. The pH and oxygen will be decreased to 7.2. And how it impact? Because it is 7.2, what it means? What it means, at least animal in the ocean cannot form the calcareate of the calcium in their body. For example, the coral larva, because in this area, you can see we are Southeast Asian country. They are triangle of the coral and more biodiversity. And this same area, we also found the hypoxia, and currently also found in the Jakarta Bay as well. That is why I come to here and alarming everyone that even eutrophication and hypoxia appear, but these consequences result is quite silent. No one interested in it because they are not appear often, but they are beneath the eutrophication. We can see the green water. So what we are doing now, if we want to do the solution, we need the social culture issue, political, environmental, and economic work together. And in here, this is the framework what I have adopted from the Xiamen University, and they bilateral in the co-partner frame under the COSO SOS. What is the COSO SOS? The COSO SOS, that is the program under endorsement of the UN decade. We're working from the 2011, 2030, working about the human activity and coastal climate change, specifically for the eutrophication and hypoxia, that we want the people to integrate the science to the management, to the social side, to support and tackle our coastal zone eutrophication and hypoxia problem. And in here, when we working, as I mentioned before, eutrophication and hypoxia, it complexes the problem. So we need co-design and engagement, not yet only from the academic institution, but we need foundation, we need enterprise, industrial, and also NGO work together. And after that, we can go to the natural side and natural bed to tackle eutrophication and hypoxia. And in here, we have the capacity training program. Everyone can join. We can share the knowledge. We can share the platform. We can share the data bank and also new technology. And in here, we will work together, not work alone. And in here, in the East Asian country, many institutions in the East Asia work together, and many platforms. If you can see that it's from the mainland China, and also from some from the Hong Kong, from Thailand, and also from the Philippines, also join together. And in here, is the site study flag of this program. They are mostly consisting in the Southeast Asian, and soon we will launch for the Jakarta Bay as well under the collaboration with BRIN. And so this is the total of my presentation. I just would like to say that, and all of the environmental issue is important, But if we not able to breathe, that is paramount issue. So this is totally of my presentation. I would like to say thank you again for host who invite me to share what I had experience, and I hope fully for the future I can have the contact with you all, particularly for young people. If you are interested in to study for eutrophication hypoxia, no matter you are biologist, you are physical, you are modeler, you are a marine chemist, please feel free to contact me or our program at this email. We are happy and very welcome to support you all. We have not only the platform, but we have the funding. We have opportunity for the master and PhD student, as same as with the postdoc as well. Please feel free to contact me.
Okay, thank you so much for the presentation, Kiki. That's such a great opportunity as well. So, for the speakers, feel free to send your slides to the chat box, as many people is looking for the slides. So now, thank you, Kiki, again. We are going to the discussion session, question and answer. So first, we have one participant ready to have the question. Mareta, Mareta's here.
Yes.
Yes, Mareta. Please feel free to have the question either for Dr. Sulis or Kiki.
Okay. Thank you for the opportunity. My name is Mareta. I'm from Marine Science major, Brawijaya University. I have a question for Mrs. Sulis, and sorry if the question is a little bit like, you explained that in the Raforasi study case, there was a significant increase in nutrient values in June, July, and August, if I not mistake. And is there a reason why this happened? And are these nutrient values is 100,000 from the result of the modeling, or you analyze the nutrients in the laboratorium, or how? Thank you.
Okay. Yeah. Yeah, Dr. Sulis, please feel free.
Okay, thank you for the question. So actually, what I showed earlier is I show that is the increase in nutrient concentration. So I also have comparison with the observation, but I think I didn't show it here because I have really limited observation data. That's why. So the location is not exactly at the Arafurasi, but I compare it with in the Bandasi location. So what I want to explain is that with the modeling, we can see that during the upwelling season, there's an increase or there's an upwell nutrient from the lower layer to the upper layer, as I showed earlier with the vertical plots. But for the observation itself, I don't have the data in the exact location like I showed before, but the location is a bit in the Banda Sea region. This is from from the cruise I think yes from the cruise data. So that's I think I hope it answer your question.
Okay. Thank you very much for your answer, Miss.
Okay.
Um, okay, for next, we have a question from Dr. Iis to Dr. Sulis. She has a question related to the if we don't have one of the input values, can we still running the models or simulations?
Okay. Yeah, thank you, Mbak Iis. So so basically models can run with any value that we use actually. So in this way, if we don't have one data set here, for example, we can simply just set the value into the constant value. So in this way, the model can still be running and we have the output. So I think this is what I mentioned earlier, that this is I think the most challenges process, that we have to provide a lot of input data and also for the boundary data. So in this way, yeah, we can still run the models, but yeah, we can just simply set the value into the constant value. I think that is okay. In EQUAM, it's possible to do so, to set the variable value into constant.
Okay.
I hope that answered the question by Is.
Okay. Is it answered? Okay. And for the next question, I think we have from Burionesti. If you want to have a question directly, we have two questions, both for the speakers, for Mr. Sulis in the chat box, for Dr. Sulis, sorry. So, first question, would you explain more about what lead the uplift nutrient during southeast monsoon? She kind of missed the information. Would you like to explain more about that, Dr. Sulis?
Okay, thank you very much, Miss Afrionesti. She's my friend actually. She was my friend in the bachelor degree. Okay, so I think is this okay to me to share maybe one slide again?
Yeah,
Show you,
feel free.
yeah, just to answer the question. Okay. So this what I mentioned about the uplift nutrients. So the previous study by Chem explained that they are under current circulation in this area. What we mean by undercurrent, it's that is where, so the undercurrent, which means that the current in the subsurface, not at the surface. So in this way, at the surface, so the current moves to the west, indicated by the green color here, from the surface up until maybe 100 or more. So because of this westward current, the water mass at the surface is moved to the west. So in this way, we have to fill out the emptiness of the mass at the surface. There are the undercurrent which move to the upper layer, like this one. So this current moves to the east, and so in this current brings the nutrient from this area. It's the deeper along the continental slope area, which I mean around this area, to the upper layer. So this is the mechanism that brings the high nutrient concentration from the subsurface to the surface. Yeah, I hope that answered your question.
Okay. Okay. Bu Rio, are you? Oh, okay. So for the next questions, we also have Bu Rio for Kiki. It is in the chat box. Based on your research, how long the duration of the atmospheric disturbance will lead to the hypoxia events? That's interesting.
Actually, for the our science study, that it's quite difficult to answer how long it is because it's a quite new science study. But one of the historical data that if we are observed in the river, that is the kind of the persistent along the seasonal during the southwest monsoon. So between the heavy rain, about 3 or 5 months, it should be the persistent in the river. But if come to the coastal line, that is quite more challenging for us to answer how that it is atmospheric disturbance. But one of our case study in 2019, when we observed hypoxia season, that we found that only one day physical change and climate intensity change at that case is a kind of the storm during our cruise occurring, that is kind of the Detroit, the stratification, that it means from the 1 day period until maybe 2 or 3 weeks. It depends on the resident time. And at the previous study, they mentioned that the upper Gulf of Thailand quite have the long resident time during the hypoxia season, maybe 39 days, because that area is a semi-enclosed sea. So it take a quite long. So if we're talking in the shallow system, the physical process and variable of the weather that is really important in terms of oxygen mixing between surface and bottom. If in case in another area, for example, in the Pearl River in China, one of the large hypoxia system, maybe take a 13 day. It quite depend and very depend on topography, water depth, and also nutrient input. And more important that is how about the physical and climate setting. But totally, in general, they have the 1 day period, day and night, but we not really call that it a hypoxia because it's normal process, 1 and day process. And some area will be weak and some area will be longer. That it really depends.
Yeah, that's very interesting, Miss Kiki. Okay, I will remind for the speaker to put the slides on the chat box because many participants asked for the slides. And then I also want to remind about the surveys for future webinars. Please open the chat box and click the Google form for the survey. And yeah, if everyone still have any question for both our amazing speakers, please feel free to. We have two more. Two more questions. First, Umir Paputungan to Ibu Sulis. First, is it open source and usable for everyone related to the models? And can we use it in the coastal area?
Okay, thank you, Pak Umir. Okay, so the first question, is it open source and usable for everyone? It's actually, this is open source, but it's not really open source. I would say that it is limited distribution among the person who has used Hamson before. So I get the code from my supervisor, but actually there is no. So ECOM is really how say it, like it's really dynamic. So it depends on the developer. They always change it, and there's no established platform like ROMs, for example. So every user get the update immediately. But as for EcoHam, we have a really limited community. So every update is circulate only for this community. And for the second question, can we use it? Yes, of course we can use it in the coastal area, but we need some how do we say it? Especially for hydrodynamics model, because for the coastal area we need a very fine grid to represent the result very well. Here, for example, what I did, I simulate a really like regional area, so the grid itself is quite big, like 7 kilometers or so. If you want to see the process in the coastal area, maybe we have to really have a really fine grid like in the order of maybe 10 or 100 meters. So I think, yeah, it's possible to do the simulation in the coastal area and use the model as well. Yeah, I hope that answers your question, Pak Umir.
And then we also have another question from Pak Dominic.
Okay.
So, based on the research, does upwelling induce any hypoxia, or if there is any cases related to that kind of situation?
Okay. I think in my cases, what I study, so in upwelling area, I haven't found any hypoxia, but it happens if you see that my area is located near in Papua area where there's a really large river, like a Diggle River there. So in my case, when I simulate the model, there is a really, really low oxygen level in this area. So it's not really in the upwelling, but in the upwelling area, but it's more like in coastal area, really close to the river area. That's what I found in my case in Arafura. So I don't know if there's another maybe researcher that have found this kind of phenomena, but in my case in Arafura, the hypoxia is found really near in the estuary in the river mouth in Papua.
And then we also have one last question, I think. Tanjung Pura University for Dr. Sulis. The question is why we use NCEP and water gap data for simulation use. Does the model resolution not affect the resulting model results, like small features such as mixing or adding in the area?
Okay. Yeah, thank you, Pak Apriansha, for the question. So what I using NCEP? So actually I had two options back then. I have NCEP and how do we say it? Not HM or F, the other one, ERA, sorry. ERA 5, back then was ERA interim. And I checked, so before I do simulation, I check like this sensitivity test, like for the hydrodynamics modeling first. And for my area, it is better to use NCEP. And also for the water gap, it's simply because because that's the data that we have, and it also has the global coverage. So yeah, we use some adjustment, and also we need to adjust the grid into our model. So there's a lot of step actually. So regarding the grid, we realized that NCEP is quite like a really low resolution. But in this way, we only focus on the general circulation, so we're not focusing really on the small scales like maybe like turbulence or so. So that's why we still using these two data set as the forcing for the hydrodynamics. Of course, if you want to see in more detail, like here, for example, in the coastal area, maybe, yeah, we need a more finer forcing data. But in my case, because the region is quite large, and we see more in the general pattern, not in the small scale, yeah, we use these two data sets. I hope that answered your question, Pak Priyansha.
Okay. I think we finished the Q&A session for now. But everyone, if there is still any question for Dr. Sulis and Kiki, feel free to just contact them via email. And then I will also want to remind everyone to put the surveys in the chat box and click for the Google form. And for the upcoming webinar, we have two speakers. The first one, Dr. Iis Triyulianti, and she'll talk about the ocean's roles in the global water cycle. And the second one, Dr. Kuo Hong Wong, and he'll talk about the importance of dissolved organic matter in marine chemistry. It will be held in May 15, 2025. So please come again for our upcoming webinars. And yeah, for now, I think we'll wrap up for our sessions. I apologize for the 10 minutes from the our supposed to finish. I hope it's
Miss Ita, sorry, can I maybe it's
yeah.
because someone asked for the material. Is it okay if I send it to the committee? Because I have to change some parts of my material before I send it to everyone. Or maybe
Yeah.
I should send it in Zoom right now, or is it okay to send it later to the committee?
I think it's better to change it first, so you will be more comfortable once you're sharing it. Is it?
Okay. Yes, so I will share it to the committee then.
Yeah. For some of the participants looking for the materials, please contact please contact us. Feel free. So we'll deliver the materials directly or contacting Dr. Sulis directly via email. So it will be more easier if I'm not mistaken. Yeah. And then thank you so much for our speakers today, Dr. Sulis and Kanita. It's such a great presentation. We enjoy a lot and we have a lot of participants for now, even until 50 participants. It's such a great webinars and we are happy to have both of you guys. And then thank you so much, participant. Yeah, for now, I'll close the webinar series with Alhamdulillahirobbil Alamin. So, thank you everyone for coming to our webinar. See you for the next webinar, number 8, in 15 May. So, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Thank you very
Thank you
much,
for all
Mbak
you.
Ita, Pak Aan, Trike. Thank you very much.
Success. Congrats. Izin live ya.
Yeah, thank you so much for coming.
Oke. Mbak Ita mungkin saya kirim lewat email aja. Kemarin yang kontak saya ada Mbak Ida, mungkin nanti via Mbak Ida bisa di-share apa materialnya yang tadi.
Boleh.
Oke.
Ida di sini, tapi
Oh, Mbak Ida
maaf.
di sini ya. Ida. Ya nanti kirim ke Mbak Ida aja ya.
Iya,
Soalnya ini
enggak.
kayaknya ada beberapa yang perlu saya sesuaikan lagi kalau misalnya mau di-share publik gitu.
Yeah,
Yeah.
nggak apa-apa. And also for Kiki, it's also okay to adjust your presentation as you wish. And after, like you feel that it's okay to share to everyone, yeah, I will share that version. So yeah, do not take a burden for this. Don't worry. Actually, I share the link of our COSO SOS program that is including for the publication and event, so everyone can enjoy. It totally is same with my PPT. So I think we can please feel free to enjoy in our website. Yeah. And also don't forget to contact me if you have any suggestion or help. I'm really glad to help. Okay, thank you very much, Kiki. So I will put the link. I opened it like several minutes ago, and it's really great and informative. So is that okay if I put that link on our description for the
Of course, yeah.
yeah.
Please,
So
please. You
yeah,
can
yeah.
share.
So
We follow
we will.
more of our document over there.
Okay, that's nice. Thank you very much. Great. So I will just put that on the description so everyone who wants to re-watch our discussion can just go to the YouTube and download it. And yeah, that will be easy for everyone. Thank you very much.
Thank you.
Yes. Thank you. Have a good day, everyone.
Have a good day.
Thank you.
See ya.
See ya.
So, jangan ditutup dulu. Enggak, maksudnya saya masih mau ngopi yang dari Mbak Kiki. Mbak
Update.
Sulis,
kalau mau apa nggak lewat email, maksudnya lebih mudah lewat WhatsApp juga
WhatsApp aja
nggak
ya,
apa-apa.
nanti via
Mm-mm.
WhatsApp aja.
Ke
Oke,
Mas Isal atau ke saya
oke,
boleh.
boleh. Sip. Terima
Sip,
kasih
sip, sip.
semuanya,
Iya,
Mbak Ida
sama-sama.
lagi.
Terima kasih banyak.
Saya izin live dulu.
Kasih.
Ya.
Hmm.